Okay, why don't we get started? Welcome back. I didn't know how many I was going to see this morning. Okay, so we've got a fair amount to cover today. The lecture is focused on fibrous proteins, but we're going to back up and finish up what we didn't get to at the end of lecture 4, which is the reverse genetics, and then we'll get into fibrous proteins. So with reverse genetics, where we left off was we're purifying some protein of interest. And in reverse genetics, really what you want to do is you're starting from a protein that you've purified. You're going to get some protein sequence, which is going to give you the gene. Once you have the gene, you can then make other mutations, put them back in the cell, and test the effects. So we left off different ways to purify proteins. So we're going to go through the Edman degradation. After you get some of that sequence, you reverse translate. And some of this is going to require giving you a little bit of advanced or early view of translation, which you're going to get later in the course. But with the amino acid sequence, we can get some information about what the DNA should look like. We synthesize small pieces of DNA, oligonucleotides, to be used in a couple of different ways to go and fish the gene out. And then once you have that gene, you can sequence the entirety of it, look at it, and say, yes, this in fact coincides to the protein that I've purified. And there are a couple of ways that you can validate that. Okay, so Edman degradation. The important point here is the sequencing of the protein is done sequentially, so that there's a free amino group here that is absolutely required to be derivatized by PITC. So this amino terminus can come from one of two places. One would be the very beginning of the protein. Another would be that you can purify the protein and then break it up into different fragments, right? Either using chemicals or some of the proteases that were in your notes, and then take those fragments and sequence them. But for demonstration, we'll just start where we're looking at trying to get some sequence from the beginning of the protein. So this free amino group undergoes a derivatization, and then there is a cleavage reaction, which is a hydrolysis. And so what you're left with is the protein that you started with minus one amino acid, and that amino acid that it's missing is the one that was most N-terminal. This derivative, this PITC derivative of an amino acid, can then be extracted from the reaction. It's analyzed chromatographically. All of the 20 amino acids with their PITC derivatives are known what their properties are, so that we can say, all right, amino acid number 1 was methionine. Then the machine goes back and does the second cycle. And amino acid number 2 comes out as being identified as whatever, all right? So this degradation is sequential in that you're derivatizing the N-terminus, cleaving it off, and saying, what is that amino acid? Go back, do it again. Go back, do it again. Okay, so a little bit of an overview just on some basics of the genetic code, transcription, translation. So you know DNA is transcribed to messenger RNA. It's the messenger RNA that is then translated into proteins. So when the ribosomes come to a messenger RNA and they start at the beginning, they're going to look at that messenger RNA. How many of you are familiar with this? Okay, most of you, so it's just a brief overview. You'll get more of this later in the course. So the ribosome is looking at the messenger RNA in sets of 3 and saying, these first 3 bases, what do I see? That codes for a certain amino acid. So it'll start with that, it'll move over, look at the next 3, etc. All right? So these are the 3 base codons that code for the different amino acids. And there's 2 important points. One is when you see U's here, that's because these bases are in the messenger RNA. There are no T's in RNA, right? Wherever you had a T in the DNA, uracil is being put in the RNA. The third point, and this becomes important when we talk about making oligonucleotides to go and fish out a gene is that at the third position, especially, these codons are degenerate. I don't mean in a bad way, in a good way. So, for example, proline, you could have C at the first position, C at the second position, and then the third position can be anything. So this would be the third base, or it's also known as a third base wobble or degeneracy, in that you could have anything there, and as long as you have two C's as the beginning of the codon, that's going to be proline. All right, so schematically, the ribosome is here. It's seeing AUG at the beginning of the messenger RNA, and it knows this is methionine. So the tRNA is carrying methionine, comes in. It'll then look at the acceptor site and say, my next amino acid is coded by GUU, which is valine. So a charged tRNA that has valine on it will come into that site. The peptide bond will be formed, and it'll move forward one. It'll look at the next three bases and say the sequence here in the RNA is GGC. That codes for glycine. So charged tRNA comes in, that binds to that, it recognizes it, it's carrying glycine, you get the second peptide bond formed, etc. All right? This should be review for most of you. If not, go back in the notes. Okay, so let's get an example. We've purified a protein, and we want to know who is it, what's the gene look like. So the Edmond procedure, let's say we go through and we get sequence for the first 6 amino acids just for the sake of demonstration. We'll then use that codon table and say, what are the oligonucleotides? What are the bases that would be in the DNA to give rise to that protein sequence? So we know methionine, ATG, is our start codon, right? So AUG, which is ATG in the DNA, that has to be there. There's no degeneracy. The second amino acid is tryptophan. Tryptophan has only one codon, UGG, so that means in the DNA it has to be TGG. Now we come to cysteine. Cysteine is UG and either U or C in the RNA, which would be TGT or TGC in the DNA. We don't know, right? At this point, we don't have any nucleic acid information. We just know that we have a cysteine there. So if we're making an oligonucleotide, a synthetic DNA, to try to go and fish out the gene of interest, we know that we would have T and G in those first two positions, but then that third position, right, it could be either C or T, a pyrimidine. Here, the next amino acid, we know the first two are GC, and then for alanine, when we look, alanine can be anything at that fourth position. So that's N. The next amino acid, glutamine, CA, and then R, meaning either A or G, a purine, etc. So when you look at that, can we synthesize one defined oligonucleotide that's going to correspond to that protein sequence going and fish out the gene? No. So what we have to do is these synthetic DNAs are made, synthesized by a machine, and we have to program it such that we have degeneracy in the oligonucleotides. So before I even get to this, let's just look back here. Let's say that was one sequence. It was a specific sequence. How many oligonucleotides would we have to make? One. If everything was non-degenerate except for a pyrimidine there, how many would we have to make? Two, right? Let's say there was no degeneracy there or there, but we had this and we had that. How many would we have to make? 8, right? We'd have to make 2 flavors of cysteine and 4 different flavors of alanine in conjunction with one of the 2 cysteines. So when you look, you'd say, well, I'm going to have to make 2 for this, times 4 for that, times 2 for that, times 2 for that, and that is how many? 32. So if we synthesize 32, question? Yes, the degeneracy is the variability at a certain position. So what you do then is when you're synthesizing these oligonucleotides, in fact, you synthesize them, the machine synthesizes them from the 3' backward to the 5'. But you would start out where you could have one of two bases here, and then you tell the machine, put in A, put in C, and then for here, what would you tell it? Put in 25% A, 25% C, 25% G, 25% C. Then we'd have, I'm sorry, that's for the N, right? For the R, it'd be 50% A, 50% G. So that statistically, we're going to get all 32 of those oligonucleotides made in there. I have a quizzical look. Didn't get that? All right, well, it's just at every position. If we know there can only be one base there, we tell the machine only put in one base. If it can be one of two, the machine will put in 50% of one, 50% of the other. So during the synthesis, half the molecules there are going to get A, half of them are going to get G. So that would give us 32 different. So when we take this out of the machine, we've got a population of oligonucleotides, and there are 32 different species in there. All right? So what do we then do? So we've got, let's say, this mixture. We can take that population and radio label it. There are various ways that this can be done, but a simple way would be we already have a library of DNA clones. We already have the DNA from whatever organism this protein was purified in DNA circles, in plasmids. They've been put into cells, and we can actually take a radio-labeled oligonucleotide and hybridize them, okay, either to the straight DNA or to the cells that have been broken open in colonies. But due to that complementarity, right, the right gene is going to be hybridized to how many oligonucleotides if the experiment's done right? One, okay? We then will have that clone isolated. We'll determine the sequence of the entire gene. If it's a human gene, we can obviously take a little bit of it, put it in the database, and say this is the rest of it. But let's, for sake of argument, say it's coming from an organism where the entire genome has not been sequenced. We need to sequence that entire gene. When we do that, first of all, we're going to make sure that somewhere in there, and in this case it was at the beginning of the protein that we got the peptide sequence, that the amino acid sequence of that gene is in fact the same as what we determined in our protein sequencing in the Edmund. Then we sequence the rest of the gene. How would we know that that is in fact the gene that we're interested in? In other words, there could be a number of genes that start out, right, with those 6 amino acids. But we know that the gene that we're looking for came from the Edmund sequencing of a protein that we had purified, and we know how big that protein is. We may know some of its properties, right, whether it's more basic, more acidic, what its isoelectric point is, so that when you get that entire gene, if you look and you read through and you translate that DNA to what the protein sequence should be, if it says this gene looks to encode a protein of 110 kilodaltons, 110,000 daltons, and the protein that you purified is 110 kilodaltons, that makes sense. If it comes back and says this gene encodes something of a different size, dramatically different, then you know you may have picked up something similar and you have to go back. Okay? All right, so are there any questions on that? Most of that should have been question.
The retranslated protein, would it code the same way as the original protein, or would it differ in coding?
Okay, when I say retranslate, in the case where I just did, that's a mental translation. It's looking at the DNA and saying, and there are programs for doing that. You give that sequence and it will tell you what the protein sequence is. It'll actually tell you what? It'll tell you the protein sequence, but how many protein sequences would it give you? If you took a piece of DNA and you got that sequence and you put it into one of these software programs and says, tell me the sequence of the protein here, how many sequences does it give you back? Huh? 3. It has a sequence, but the computer doesn't know what is the reading frame, right? So you've got a sequence, and remember a codon is 3 bases. So in a particular region, you have a whole series of bases. Do you take this 3, or do you move over 1? What is the register? Okay? If you think about that in a cell, the messenger RNA is made, and you know the beginning of the message is what starts the register. It's the first 3, starting from the beginning of the message. But when you're looking at a DNA sequence and you don't necessarily know where is the beginning of the message, then the software will look at it and says, well, if you start here, then we'll look at sets of 3. And maybe it only goes a short way, and then it hits a stop codon, which means that's not going to make a protein. There's several of the codons that tell the ribosome to stop. So it may say, well, if I start in this next register, not the first set of 3, but I'm going to move over 1, take that set of 3. That's the second reading frame. And that may give you what's called an open reading frame. It'll give you a long protein or predicted protein. Okay, so let's go on to fibrous proteins. So over the next few lectures, we're going to start to take what we've learned about protein structure, properties, and look at some examples. And we will look at enzymes, proteins which have a globular, 3-dimensional structure, will carry out chemical reactions. They will act as protein catalysts. But for fibrous proteins, they are not enzymes. So the proteins we're talking about today are not enzymes. Their structure is, in fact, their function. So these are structural proteins. They're providing some structural assembly to the cell. They are structural proteins, structural fibers, all right? They're usually rod-shaped. They're not globular. And they have secondary structure, but they don't really have a globular tertiary, where we talk about folding up. Here, you'll just see several examples of this higher order packing of the secondary structural arrays. So you have a secondary structure that's made for one molecule, then another molecule with the same secondary will come in, interact with it, and they will pack together to make something that's important, but they don't really fold into 3D structures. We'll see that a common theme in them is that they have a repeating motif or sequence unit which gives these proteins some periodicity, and you'll see how that comes into play with respect to packing. And in some cases we get these special modifications, covalent bonds, to allow these associations to occur. So we're going to talk about the alpha keratins, which play important structural roles for hair, wool, nails, fibroin, which is the structural protein for silk, and then collagen, which is the #1 protein in your body. All right? It is for tendons, blood vessels, skin, bones. I think 80% of the protein in your body is collagen. Okay, so before we look at the specific proteins, we can look at a composition table. Remember, we can determine the composition of a protein simply by hydrolyzing it, right, in acid. Now, let's say you took some garden variety protein. You looked at all the proteins, and on average, you say glycine is present at about what frequency or what percent in the general population of proteins. You'd say, yeah, it's usually there about 8%. That is typical. When you look, you see a number of instances for these different amino acids where they are very much enriched in the fibrous protein that you're talking about. These fibrous proteins don't have the full spectrum of amino acids being represented, but they're really made up of relatively few number of them that are in these structural motifs that are repeated many times. So when you look at, let's say, alpha-keratin or fibroin, you look at fibroin, 44.6% of the protein is glycine. Nearly half of it is glycine. Collagen, 33%. Alpha-keratins, you look, cysteine is 11%. In the general population, it's about 1%. And what do we talk about cysteines, what they can do? Disulfides. So the point here, and you can, after we look at them, you can come back here, but really you have a very restricted composition, bless you, of these proteins with respect to the amino acids that are represented in them. All right, so alpha-keratins. First, they're essentially all alpha helical, and with respect to that motif that we're talking about, there's these proteins have some repeating unit. For alpha-keratins, we call it a 7 residue, and somebody had been confused before when we say residue, we're also saying amino acid, right? A 7 amino acid pseudo-repeat, bless you. So a pseudo-repeat is you don't have, let's say, 7 contiguous amino acids that are perfectly repeated over and over, right? Pseudo-repeat is the repeats look pretty close to one another. There's some variability, but in fact, within that 7, what is rigorously conserved is that positions A and D are hydrophobic amino acids. Okay? That is very important for understanding the structure and then the properties of the alpha-keratins, and that in that 7 amino acid pseudo-repeat, position 1 and position 4 are hydrophobic. In an alpha helix, what did we say regarding the helical turn? How many residues per turn in an alpha helix? 3.6. So if you have, and we talked about the helical wheel projection, if you have a hydrophobic residue here, right, and then you move around another 3, you're going to have another hydrophobic, and it's going to be almost lined up with that first one, right? So if every A and D are hydrophobic amino acids, what you're essentially going to get is something like this. Here you have, let's say, one of those alpha-keratin monomers. And you have A, right? A, B, C, D. So it's 3 residues, not 3.6, so it doesn't quite come all the way around. But on this side, you now have 2 hydrophobic residues. When you go around another 3, right, it's going to be, let's say, down one rung of the ladder or up one rung of the ladder of the helix, but it's also going to be a little bit staggered. So if you look at this, okay, you're basically going to see that there's a strip of hydrophobicity that's on the outside of the helix that's going with the helix. Okay, does that make sense? All right, So that's on one monomer. Now another monomer has the same structure. So what can those two do? They're going to line up, and in the middle, where they're twisting around each other, what are the surfaces of those two monomers that are making contact? The hydrophobic strips. So here, the sequence is imparting the properties. You've got this repeated 7 residue where 1 and 4 are hydrophobic. That's then giving you an alpha helix that gives you a hydrophobic strip. 2 of these monomers come together and twist around each other. All right, and why are they associating? Hydrophobic effect, primarily. So when you have 2 of those monomers, they'll come together and form this structure here, which is called a coiled coil, because one monomer is a coil, alpha helix in this case, and 2 of them are coiling around each other. So it's a coiled coil where 2 of those monomers come together. You then get, which is characteristic of these fibrous proteins, higher order packing. So here's a coiled coil, here is a coiled coil. We said that those hydrophobic residues are conferring the association, the intimate association between the 2 alpha helices, but there are other residues there, right, that are not buried in that hydrophobic strip-strip association. Those on the outside can associate with each other to bring 2 of those coiled coils together to pack up to another structure known as a protofibril, and then they pack in bundles. And when they pack in these bundles, they're called microfibrils, and then those can pack to give even bigger fibers, which are macrofibrils, and that is a depiction of, let's say, a cross-section of hair. So that in the hair, you just have these bundle fibers, okay, that are these repeating units of the alpha keratins. So what about the cysteine? The cysteines can form disulfides, and there is disulfide formation, which is going to stabilize these fibers, all right? And the amount, what did we say? We said alpha keratins are in hair, wool, fingernails. It's the same structure. The difference between, let's say, your fingernails and your hair, one of the differences is there's a much greater extent of disulfide formation between these fibers in your fingernails, all right? If someone wanted to get a perm, you probably know it's a smelly deal, right? But what's really going on here? When these fibers in the hair, they have normal disulfides that are holding them together. So the first thing you would do is, I want to break those disulfides. So how is that done? It's reduction so that the disulfide bond are broken, and now you have 2 free sulfhydryls. Now what do? You take the hair in that state and you roll it. And when you roll it, let's say you had fibers like this, they're going to move relative to each other. You're kind of stretching the hair a little bit. Then what do? You neutralize that and let the disulfides form again. And so now you've got a shift, and now cysteines that were previously bonded with somebody else are now bonding with each other. You've got a shift. So you're breaking disulfides, changing the position of the filaments, allowing new disulfides to form, and then that's going to hold, but it's quasi-permanent, as you probably know, because eventually you can get reduction, reoxidation, and basically the curl will fall out. Okay, so fibroin, major component of silk. In this case, we're talking about an anti-parallel beta sheet as being the secondary unit, or the secondary structure. The repeating unit is serine, glycine, alanine, glycine, over and over and over again. In a beta chain, a beta sheet, beta strand, what do we know about the R groups as you go from one residue to another? Up, down, up, down, up, down. So serine and alanine are going to be on the same side or opposite sides of the beta sheet. Same, right? Up, down, or down, up, down, up. Glycine, they're all going to be on the same side. Glycine, what is its side chain? It's important for this structure and for the next one. H, it's the smallest side chain. So what we'll see is that these side chains between adjacent sheets are alternating big, small, big, small. First of all, alanine and serine are not that big, but they're bigger than glycine. They're not charged, they're not big and bulky, but they allow these sheets to interdigitate, as you'll see. When we look at silk, the fibers that are formed, because you have beta sheets, those beta sheets can move, so silk is kind of flexible, but relative to an alpha helix, a beta sheet is more extended or less extended? It's more extended, so you can't really extend it further, so it's not very stretchy. But it is flexible. So your notes show this. So let's say we have two sheets coming together. The glycine is here. Then you have either an alanine, which is, or serine here, it's showing an alanine. So all of the glycines are going to be on one side of the sheet. All of the larger R groups, either alanine or serine, are going to be on the top. So that's shown kind of like this. You then have another sheet that's going to come down and pack on top of it, and you're going to get this interdigitation. So let's say when you look at this sheet, what we're looking right there is how many strands for each sheet? 2. So if you look here, you can see that there's a pocket, right? There are no R groups projecting up here. The glycines are pointing down, so these two from this 2 strands are going to be able to fit into there. These 2 R groups from this sheet are going to be able to fit into the pocket of this sheet because the small glycine side chains are projected up, okay? So you have this kind of an interdigitation which is allowing these sheets to stack, all right? Again, no 3D structure per se, you're just getting a stacking in a higher order array of those sheets. Okay, collagen, #1 protein in your body. We have a repeating unit, which is glycine, then something, all right? Very often, the X is proline, and very often the Y is hydroxyproline. So is hydroxyproline one of the 20 amino acids? No. So it comes about by a post-translational modification, all right? It comes about by a modification of an amino acid after the protein has been made. The important point here is that every third amino acid is glycine. All right, it has secondary structure that is helical, but it is not an alpha helix, as we'll see. All right, it's not an alpha helix, it's a left-handed helix, and it only has 3 residues per turn. So if an alpha helix, if you go 3.6 per turn, a collagen helix is left-handed, okay, and that has only 3 residues per turn. So is that more extended or less extended? It's more extended. You're only getting 3 residues as opposed to 3.6. There are no intra-chain hydrogen bonds. We talked about the fact that in alpha helix, right, we have the hydrogen bonds that are stabilizing that secondary structure. Here we don't have these intra-chain hydrogen bonds stabilizing that left-handed helix, but we will see that there are hydrogen bonds that are occurring between strands, right, between these units, all right? Okay. So our monomeric unit for collagen is a left-handed helix. The next order of packing is tropocollagen. So that is a helix of 3 helices. It's a triple helix of 3 left-handed helices, but this is a right-handed superhelix, right? 3 left-handed helices come together in a right-handed fashion and give you what's called tropocollagen. So I already told you that there's no hydrogen bonds within one chain, within one left-handed helix holding it together. But if we took this tropocollagen, let's say we sliced it, took a cross-section, and we looked at it. In the primary structure, what amino acid is present every 3 residues? Glycine. Glycine has the smallest side chain. And the reason, all right, is that as these helices come together, we'll call this strand A, strand B, strand C. Here's a cross-section, and when you look, strand B, strand A, strand C, and you say, what is projecting to the interior? What is projecting towards the axis of the superhelix, where these 3 helices are making the tightest contact? It's glycine. So that's why you have glycine every 3rd residue, is to minimize steric hindrance between these helices coming together. So you have these hydrogen bonds that are stabilizing between the strands, all right? So then what happens? Like we saw for the other fibrous proteins, we get higher order packing. So in this case, we have the 3 strands coming together, and here is a model of that. These fibers, now the tropocollagens, they're going to line up, and then right next to them we'll get another set of the tropocollagen lining up, but they'll be slightly offset. Okay, so this arrow is showing you here's a tropocollagen, here's a tropocollagen, there's a tropocollagen. So how does that stay together? The way that they stay together is that we start to get cross-links. Cross-links between adjacent tropocollagens. So if we get a covalent cross-link, a chemical bond between this strand and this strand, these 2 are then going to be tethered. If we get one between here and here, these 2 are going to be tethered, which means this is tethered to that, relatively speaking, because they're both tethered to that other fiber, right? So this covalent cross-linking after you've lined these tropocollagens up are what hold the whole thing together, all right? And you get these fibers. So if you look by EM, you see these striations. Those striations correspond to the gaps in terms of the lining up of the tropocollagen. Any questions on that? Okay. So how does that cross-linking occur? It involves the side chains of lysines. So there's an enzyme, lysyl oxidase. So what I've shown here is the side chain of lysine. The lysyl oxidase will work and oxidize this side chain and give you an aldehyde group. You don't have to worry about the chemistry, but you should know the basic thing that we're trying to achieve here is that we're getting a cross-link between the fibers. This enzyme is carrying it out, and it contains a copper ion. And we'll see other examples where things are working in enzymes, right? The presence of a metal ion, a metal atom, and how it participates in the chemistry. But at this point, all you need to know is for this enzyme to do what it has to do, it has to have a copper ion associated with it. When you have this aldehyde, then a lysine from an adjacent tropocollagen fiber will come into proximity unmodified, and you'll get this bond, right? So it's not a disulfide bond. It's a lysine-lysine bond that is coming about by the action of this enzyme, lysyl oxidase, and it requires copper. So that's how we get these cross-links. Okay, so as an aside, the amount of cross-linking between these fibers, as we get older, we get more and more of these cross-links. I'm not sure why. So what's going to happen? As we get older, our skin is going to become, right, less flexible, less appealing. It loses the elasticity. Our bones are going to become more brittle. So that's if you have too much cross-linking. What if you don't have enough cross-linking? Okay, then you're going to weaken this structure, and one way that would do that would be if you had a copper deficiency, right? Okay, we have a second type of post-translational modification that goes on in collagen. So the first one is involved in forming these cross-links. The second is really important for all those hydrogen bonds within the tropocollagen. So I showed you here where we're getting these residues, and I said often you'll see proline or hydroxyproline, all right? When you think about proline and you think about hydroxyproline, they differ by virtue of what? A hydroxyl group. What can a hydroxyl group do in hydroxyproline that the hydrogen in proline cannot do in the same fashion? A hydrogen bond, all right? In terms of that oxygen being introduced to make the hydrogen on the hydroxide more positively charged. So what's going on here is the first enzyme that we talked about is giving you these covalent cross-links between the fibers. The enzymes proline hydroxylase and lysine hydroxylase, they are introducing OH groups, okay? And those OH groups are now participating in the hydrogen bonds that are stabilizing the core of these tropocollagen superhelices. So they're very important for helping to stabilize that. Remember, within one helix, within one left-handed helix, are there any hydrogen bonds in collagen that are stabilizing that? No, it's the hydrogen bonds within the three helices in the tropocollagen. And for those to be there, you need these modifications. You need to introduce those hydroxyl groups. So both of these two enzymes, they require iron. And like I said, when we look at mechanisms of catalysis, we'll look at what it is that these metal atoms actually do to help out a reaction. For our purposes here, you don't need to know that, but what you do need to know is that these metal ions can be in different oxidation states. Different amounts of electrons are on them. So to maintain this iron in the proper oxidation state for it to function, you need ascorbate, ascorbic acid, which is vitamin C. So when we talk about vitamins later, we'll see that what vitamins are doing is they are playing a role in enzymatic reactions. They're not the protein per se, but they're these other more these non-protein parts of either the enzyme or helping out in the reaction, that they're helping the chemistry. So here, vitamin C is helping the chemistry because it's keeping the atom that is needed by these enzymes in the proper oxidation state, okay? So, what happens if you don't have enough vitamin C? Right. So, when you think about scurvy, what is it really that's going on? I mean, on the outside, you'll look at a scurvy dog, and their body is essentially falling apart, right? They're hemorrhaging because their blood vessels are weakened, right? Teeth falling out, skin lesions, everything. It looks like they're falling apart. And it's really because of one thing. They don't have vitamin C. If they don't have vitamin C, these enzymes that have to work to give these modifications to give the structure that gives collagen the function that it has, right? It has a structural function, maintaining the integrity of your being. But because the vitamin C is not there to keep that atom, that iron atom, in the right oxidation state, these enzymes can't function properly. They can't function properly. You don't have the OH groups. You don't have the OH groups, you don't have stable tropocollagen helices. Okay, so then it's a whole house of cards. So, who wants to answer the question? Why do they call English sailors limeys? Because once they figured out, when they put limes on board and they had the citrus to provide them with the vitamin C that they were lacking, they were able to do their jobs, all right? So what you need to know from today is, again, hold on, because I noticed at the end of the lectures, you can always hear my voice having to go up at the end because I'm fighting your noise. Know the differences between these different fibrous proteins in terms of how their structure differs, what they have in common in terms of the repeating motifs and so forth, but what the differences are that give them the different properties. So tomorrow tomorrow we have recitation and we will go over problem set 1, and then Thursday we'll start to talk about globular proteins.